No, just as I was saying, last one of the morning. I know it's tough, but I will try to be painless. So yeah, as Luca just presented me, my name is Mikel. I'm going to present you the study I've been working on for the last months, consisting on an eDNA-based approach for quality assessment of estuaries, studying their sediments. Wait. Okay, so on the one hand, we have microbes. Probably all here know they are the base of Earth's biosphere, essential to all ecosystems, since they carry out really basic functions, such as nutrient cycling, but also decomposition of a big amount of compounds, not only organic, but also human-originated ones, such as contaminants. And then, on the other hand, we have estuaries, some of the most complex, dynamics, but also biodiversity rich, as well as resilient habitats on Earth, since we humans have often decided to settle around this and build big metropolitan areas. So the impact has been notable. And the problem or the connection between these two is that often when assessing estuaries' ecological status or health, microbial communities are neglected, are ignored. And this has happened because traditionally studies assessing their health were based on macrobenthic, macro like eukaryotes. But the good thing is that since the last decades, relatively recent development of new technologies, as Professor Elena Boujan was presenting us yesterday, the so-called next generation sequencing. This, thanks to genetics, have allowed a way easier and more efficient study of microbial communities, for instance. So the good thing is that microbes can actually become good indicators of ecological status of different ecosystems, including estuaries. So the methodology or the method we have used in this study was a study of eDNA, which is environmental DNA, and it's inside the next generation sequences technologies group. And what does this consist of? Basically, we do take bulk samples straight from the environment. It can be water, soil, sediment of estuaries in this case. Then we perform the metabarcoding, which consists on the DNA extraction of the whole sample. So you take all the DNA present, you amplify it via PCR and the use of universal primers. And finally, there's the sequencing, which gives you as a result the sequences, which you will later compare with reference databases or libraries, as they are also called, which by the end you can have a picture of your community composition in the samples. You can have, you can know what was there. So all this said, the aims of this study were, first of all, to assess whether eDNA metabarcoding was a suitable and effective approach for assessing the health of estuaries. Next, at the same time, we wanted to see if either prokaryotic or eukaryotic or both reflected differences of the anthropogenic pressures between different estuaries. And lastly, again, if this methodology, this approach, metabarcoding, is also accurate when detecting invasive species. So for the methodology, I want to spend much time here, so you can ask me if you need anything later on. For our study area and sample collection, so we studied 6 different RIAs in the northern Spanish coast. And the good thing about these 6 different estuaries is that they were pretty different in their historical backgrounds. And as you can see here, you will see these different symbols in the area of results and discussion to indicate like really industrialized or urban or natural. And yeah, as I was saying, the good sample size of this study is that there are big differences. For example, we have Bilbao is the biggest metropolitan area and also the most industrialized one, especially in the 1970s, followed by A Coruña and Vigo, also big cities and industrialized areas. And then in the contrary, for instance, we have Ribadeo, which was even considered as a biosphere reserve in 2007. So the sample collection consisted on taking sediment core samples from which 2 different layers or depth were taken. Basically, surface layer, upper layer, hoping it to be the oxic one, and then the deeper layer, or layer 3, I will be calling maybe, for the deeper one, which hoping to be anoxic. Concerning the universal primers I was talking about earlier before. We use the 16S rDNA V4 region for the prokaryotes, those are the prokaryotes, and 18S rDNA V9 region for the metabarcoding of the eukaryotes. And lastly, for analyzing all the sequencing data that we had, we used one main bioinformatic pipeline, Time2, standing for quantitative insights into microbial ecology. And then in order to get some graphical representation of the biomarkers, so of the big significant differences between the RIAs or the depths, we used LabSe, which stands for linear discriminant analysis effect size, and you'll see some results like this. And regarding the different reference databases, we use two, Silva 132 and PR2. So straight into the results. Before looking at this graph, so overall composition of microbial communities when including surface layer and deeper layer showed nothing unexpected. So upper one, oxygen dwelling taxa, sorry, and deeper layer, anoxic nature taxa. However, now you can look at this graph. When assessing the different RIAs, the different estuaries, there were some actually clear significant differences between them, which actually reflected the anthropic background of the different estuaries. And the best examples were Bilbao, again, as I mentioned before, biggest metropolitan area and also most impacted by industrialization. So here we found different taxa such as Desulfobacterota and Zeta protobacteria. Desulfobacterota, bacteria using sulfur and iron to make anoxic respiration, and Zeta protobacteria, iron oxidizing bacteria. So this one, the presence of this taxa was clearly reflecting as the presence of iron and possibly sulfur in the area. And then also significantly abundant, Campylobacterota. These ones include several bacteria of public concern, which cause especially gut diseases. And this is also a clear reflection of the big human sewage in the estuaries from the city. Then following, also, this was quite unexpected. So this taxa over here with this really weird name, Ectothiorodospiraceae. This is purple sulfur bacteria, which is actually shows a preference for really hypersaline and hyperalkaline environments, which was not expected to be A Coruña. It was expected to be polluted and impacted, but not that much. And the presence of this bacteria indicates that no other photosynthetic bacteria was able to live in that area. And probably most surprisingly, we found a significantly abundant group over here in Ribadeo, which, as I said earlier, it was the pristine area of our study. And this group, UBA 4486, was recently classified as a facultative methylotroph, meaning they can grow with the use of single carbon atom compounds such as methane and methanol, which obviously doesn't make much sense in a pristine or in a biosphere reserve area. But later with the eukaryotic results, we found that in this exuria there was an algal bloom, and it is known that several phytoplankton taxa are able to produce methanol. So we were able to correlate the presence of this group with the eukaryotic results, and that was pretty nice. So in conclusion, or take-home message of this one, bacteria do actually reflect the differences in the anthropic impacts or backgrounds of the different RIAs. Moving to the eukaryotes, it was not that easy to make conclusions for these ones, especially because it was different to differentiate between alive and dead organisms because of the metabarcoding of DNA and not RNA. And examples of this is, for example, the presence of, these are the most abundant taxa found, and there were no significant differences between RIAs. So you won't see the bar plots like before. And what I was saying about alive and dead, for instance, these two groups were abundantly found in the deeper layer, and Embryophyta, these are terrestrial lands, so land plants, no sense, obviously, that they were alive within the estuary, within water. So the presence of this group is basically deposition, so the decaying of a dead organism. And similarly, we have here Dinophyceae, which is again found in the deeper layer, and they are photosynthetic dinoflagellates. So yeah, it makes no sense that they were alive in there because they need light to survive. So again, result of dying organism and deposition and decaying. However, we could find some taxa which could reflect some patterns or reflect the different impacts. And the clearest one, the most clear one, was Stracoda, which has been found to be highly impacted by human pressures in marine environments. And in this case, you can see that there is basically no presence at all in Bilbao, and it's probably a reflection of the high pollution or contamination levels, and also the levels are lower in Vigo. And there's also other taxa for which not much is known. There's not much literature about it, but they show some patterns that could reflect these differences in the impacts. And these are aquas like bilateral worms and platyhelminthes. So again, not much is known, but with further research, they could be someday maybe used as bioindicators, but they show some patterns of no presence in actually the most impacted areas, which are A Coruña, Bilbao, and Vigo. And same here, like really scarce presence. So with the eukaryotic taxa, it was not that easy to make conclusions, but they could become potentially good bioindicators with further research. And however, for the eukaryotes, the most relevant results we found were the presence of several invasive species. So this is Alexandrium. This is the algal bloom I was talking about in the biosphere reserve area. And they actually cause harmful algal blooms. And then several copepod taxa, which have been found to have big impacts in the whole copepod community, as well as in the zooplankton community of aquatic environments. And when using the two different databases, we could see some differences in the accuracy of detection or in the sequencing classification. So with the Silva 138, for instance, we have Acartetonsa and Aitona davisae and Calanipeda aquadulcis as the copepods. And with the Silva 138, we found Acartetonsa and Calanipeda aquadulcis, while in the PR2, we found Acartetonsa and Oitona davisae. So these are slight differences of the accuracy in the reference databases, but which are actually being developed continuously, developed and improved. But the take-home message here is that this methodology actually allowed the accurate detection of these two species, which are actually having an impact. So to finish with the take-home message or conclusions, again, we can conclude that this methodology, the eDNA metabarcoding, is actually an efficient and pretty affordable. Again, as Elena Bouchard showed us yesterday, with the years, the price of this methodology has dropped significantly. So this methodology is actually a really effective approach to assess the ecological status of, in this case, estuaries. And then we have that prokaryotes, and especially bacteria, reflect with pretty high accuracy the differences in the anthropic background of the different estuaries, while eukaryotes, some more research should be done with eukaryotic taxa, but some of the taxa could become like potential bioindicators. And finally, this methodology was actually efficient in accurately detecting several invasive species. So this was the research group I was working with, and that's me on my email if you ever want to write me. So thank you, and thank you for your attention. If you have any questions.